Okay, good morning. Welcome to Wednesday morning, the second plenary day of the conference. I hope everybody's having a good time. And we're going to continue with the first plenary session today, which is another excellent session on neuroscience. It's Cortical Oscillations, Waves, and Consciousness 2, following up on Earl Miller's keynote, which was more or less the one version of that, since a lot of this revolves around his work, or is connected to his work, not all of it. So our first speaker is remote. He's André Bastos from Vanderbilt. And when we see his slide, I can tell you that his title is Multi-Area, High-Density Laminar Neurophysiology, Madeleine. Recordings in the non-human primate brain suggest predictive coding is implemented via predictive routing. Andre is from Vanderbilt. We just saw his institution affiliation. And with that, Andre, please take it away.
Thank you. Appreciate it. The video sharing is not yet enabled. So if you guys want to be able to see me, you can quickly do that before I get started. Still disabled. So I'll go ahead and get started then and deprive you from my wonderful mugshot at the corner of the screen. So yes, good morning, everybody. It's a pleasure to be here. It's a real pleasure to be speaking at a consciousness conference, actually. My very first conference ever as an undergrad was the ASSC, and haven't been back to another consciousness conference, so this is the first time for me in many years, and I'm hoping to be able to interface much more with this topic and this audience. So here we go. So what are we doing here? We are studying the how different parts of the brain talk to each other during cognitive control, and how this is changed by propofol-mediated unconsciousness. And we've developed a model. Now, this model, you might have heard a little bit about it from Earl Miller yesterday. We indeed developed it together while I was a postdoc with him. And we're continuing, there we go, there's my video. We're now continuing this work at Vanderbilt University, where I'm an assistant professor. So this predictive routing model, it's a model for how the cortex controls its own activity, and it's building upon a model called predictive coding. So what is predictive coding? The predictive coding model has been extremely influential in neuroscience over the past, I would say, 20 years or so, maybe a little longer than that. And here's what the predictive routing model says. Okay, it's a model for how different neurons respond to different things in their environment. So the hypothesized circuit is diagrammed out here to the right. So let me explain what's going on here. The higher order cortex, typically this is thought of as higher order prefrontal cortex, issues predictions, right? So predictions are, what do you plan to do next? What do you think you'll see next and sense next? And these higher order predictions are communicated to lower levels of the cortical hierarchy, where they meet the sensory information. In this case, we're showing the visual input, right? So sensory input is then compared to this top-down prediction. And then as a result, if the predictions match the sensory input, then we're done. We don't actually have to do very much signaling of that sensory stream. But what if there's an error, a mismatch? Well, if there's an error, then there's going to be a prediction error that's generated by this circuit, and that prediction error is going to be computed in superficial layers of cortex and relayed in the feed-forward direction, these blue arrows here, where they can reach higher order areas of the brain and then create better, update those models, those prediction models, and then make better predictions in the future. Now, what's the connection to consciousness, right? So here's what I'm asking. The questions that I'm asking now of this model is the following. Predictive coding was initially back in the late 90s, 1999, and even earlier than that, the work of Srinivasan in the 80s. It was a model for very low-level visual processing. but this model has been incorporated and expanded into cognition. And so my question then is the following. Is predictive coding purely a sensory-driven computation, or does it involve higher-order computations, as I just outlined, these predictions and this model-making process as a way for us to understand our conscious experience? Okay, so there's two aspects of that story. The first aspect is, where are we going to see evidence for predictive coding? In the low-level visual cortex exclusively, in the high-level cortex, or everywhere in between? And a lot of times this predictive coding model is discussed in papers as if there is already a very high degree of confidence about it. But in fact, we haven't tested some of its basic tenets. So that's one of the things that I'm working on. And then the additional question is, if it is higher order, then we would expect that predictive coding and predictive routing, as we'll get into in a moment, processing should be selectively turned off during states of unconsciousness, which we're also going to test. Okay, so here's how we have been testing the predictive coding model. We've been testing it with a visual oddball task, and the visual oddball task creates sequence level predictions through habituation. So think about these sequences right here as basically what you see every day as you're driving to work, or as you're commuting to work, or walking or biking to work. You get a sensory flow that is very similar from day to day. And so it wouldn't make sense for your brain to use up all of its resources in processing that information, right? So in order to have a very simple model of that, what we did was we habituated, in this case, macaque monkeys, as well as mice. We've also done this a little bit in humans, but we don't have the neural data for this. So across a lot of different species, today I'll be speaking about the macaque monkey data, though. We've habituated these subjects over 3 to 4 sessions to expect nothing but the following stimulus sequence: A, B. Okay, so the A in this case is a drifting grading. We can, of course, alternate what we call A. In this case, it's a drifting grading with the grading pointed to the left. And then the animals are simply passively viewing this, A, and then B. B, over and over and over again, to create this idea of sensory habituation. This is like going to work. First you see this stoplight, then you see that street, and then finally you're at work, B. Okay? And then during the recording sessions, what we did is the following. We created a situation of what we call a global oddball. And so a global oddball is a deviation from expectation, because you're expecting B. So the global oddball trials are the A trials. And now it's important that when we test predictive coding, we don't just simply call a local deviant an B evidence for predictive code or for prediction errors. And why not, right? Why wouldn't that be strong evidence for predictive coding? Well, it would actually conflate very low-level neural adaptation mechanisms, and we've known this for over 100 years, that neurons adapt to repeated stimuli. Once you give a new stimulus, then you activate fresh neurons that weren't adapted, right? So we want to really make sure that we distinguish that level of, perhaps you could call it very low-level predictions, or you could call it low-level neuronal adaptation. We want to distinguish that low-level adaptation from a higher order process of prediction, which we get at in this particular task by manipulating the probability of the sequence as a whole, right? So in the habituation, that's all that the subjects see is B. In the main block during neuronal recordings, now they get the global oddball. And the question that remains open in predictive coding, believe it or not, is, does the sensory brain, does it get activated by these local oddballs, these stimuli? Okay, so in order to get at that, what we're going to do is the following. We're going to compare the unpredicted A to the predicted A. So we're going to compare the same exact stimulus in 2 different contexts. Context 1 is when it is unpredicted, and then context 2 is the A stimulus when it occurs later on in the task, after we've destroyed the predictions, because we've later on in this task, we would have also given the animals or the people very random stimuli. So now all bets are off. There's no longer this strong AAAB prediction. And now we can contrast that A stimulus when it is in the unpredicted versus predicted. And that's what we're going to call the global oddball comparison. And we can also, of course, look at just simply release from adaptation. And that's going to perhaps reflect a very low-level process. And that release from adaptation effect is what we call the local oddball comparison. And that's simply the unadapted B stimulus, so the AAAB, so it's still fully predicted, but it's unadapted compared to the fully adapted B response, B. Again, when it happens later on in the session, in this recording session. Okay, so again, that's the key thing here. Where does local oddball processing, very low level processing occur? Where does higher level global oddball, more sequence related processing happen? So now in order to get at that, we turn to macaque monkeys because they can be trained on this task, and we can develop a very, I think, powerful new technology in order to record from all over the brain. So that's what we did in this task. We recorded from primary visual cortex all the way to V2, V3, V4, medial temporal areas, which are much higher in the hierarchy, TEO, which is part of this temporal stream of processing, all the way up to prefrontal cortex, where we think the models themselves are housed. And so one of the things that we've been developing in my lab is this idea of Madeleine recordings, right? So Madeleine recordings are multi-area, high-density, laminar neurophysiology. Now, what do all those things mean? Well, they're all very important processes for us to be able to understand where in the brain a predictive code emerges. Where in the brain does a predictive code, where is it born? Right? Because if I would only pick one or two of these areas to record from, then I wouldn't be able to answer that question, because that question relies on our ability not to just think of individual neurons in the brain and individual areas doing one little function, but it requires a much more holistic, much more all-encompassing, and much more of a network view of the brain. So that's the multi-area. The high density refers to the fact that now we can record from hundreds of individual neurons across particular layers, and we can record both their spiking activity, which we think of as the neural code, how do neurons encode information about the world, as well as the different neuronal oscillations. So then we can, with this technique that we've been developing in my lab, we can answer this question: at what stage of processing does a predictive code emerge? So here are the areas on the brain. This is the front of the macaque brain, the back of the macaque brain. Here are the areas in their hierarchical order as established through other neuroanatomical data sets. Here is our recording technology across the different layers of cortex. So here you see a Nissl stain showing very beautifully the laminated structures. Layer 4 is the input layer, layer 2, 3 is the feed-forward output layer, and so on. In this talk, I'm not going to yet go into too many details on the layer, but you're going to see it a little bit later. Where do these different rhythms actually emerge, and what layers do they emerge? Okay, so here we go. So what's being shown here is now already the result of the local oddball comparison. So recall, the local oddball comparison is the unadapted B versus the B when it is fully adapted, okay? So this is what we would call release from adaptation. So where does this release from adaptation happen? The blue is the release from adaptation. The red is the fully adapted response. So we're not yet talking about prediction here. We're just looking at this low-level adaptation phenomenon. And what we are plotting here is the neuronal response. So this is spiking activity of many single neurons recorded in each of these areas. We've recorded a minimum of 200 to 300 individual neurons, up to about 800 to 1,000 neurons in each of these areas. And what we're doing here is we're expressing change from baseline activity, okay? So that means how much do the neurons spike to the oddball stimulus, to the AAAB stimulus, when relative to the background firing rate, relative to just their low-level firing rate without any stimulus in it, right? So that's why the baseline here is set to 0. Okay, so we can now plot the activity for these two conditions. And what we find is that this local oddball activity, this release from adaptation, it happens as early as V1, and then it feeds forward throughout the sensory processing hierarchy. One area inherits a little bit more excitability than the other, and it just continues to get processed all the way up to prefrontal cortex, where that difference between local oddballs and the control stimuli reach the apex, reach the strongest level of activity. And so this happens everywhere, and this is consistent with a lot of prior work, namely that mismatch negativity and many other studies, right? But what we wanted to know with this experiment is, okay, how about now when we actually control for the effects of adaptation and look purely at the effects of prediction? So that's the global oddball. That's this next slide. Again, now we're looking at the unpredicted but fully adapted AAAA versus the unpredicted but again fully adapted AAAA. So we've controlled for all possible confounds, including all possible effects of stimulus history, effects of motor confounds, effects of the time in the task, and so on and so forth, the neuron's individual responses. Everything has been equated except for prediction. And what we find there is, very interestingly, the sensory areas, these lower level, early and mid-level areas of the brain, they spike just as much to the predicted AAAA, which is the red, as well as to the unpredicted AAAA, which is the blue. So this putative proposed prediction error computation that many models have been very influential on, it does not occur in the low-level visual areas, okay? At least it doesn't occur to these higher order types of prediction error responses. But interestingly, we did find that it emerges in relatively higher order areas of brain processing. So definitely in these some of these mid to high level structures like TEO, as well as PFC, you start seeing a differential response to that same adapted stimulus, but now when it is a prediction error. So that's where these global oddballs emerge, is in higher order cortex. Now, one interesting thing that we can also look at, in addition to how much excitability is there across these different areas, we can also look at the brain waves of activity, right? And previous work has linked gamma oscillations, these 40 to 100 hertz oscillations that you're seeing here in yellow. It's linked that work to prediction error encoding. And so again, we wanted to ask, well, how about now that we've controlled for prediction error, we've controlled it for stimulus adaptation, now is there an effect of global oddball responses on gamma oscillations? And we found that there is. So an unpredicted global oddball, AAAA, minus a predicted AAAA, will in fact give you an enhanced gamma response, okay? So the unpredicted AAAA, and this is of course a spectral time frequency analysis here. So again, 0 is relative to the oddball stimulus relative to the A response, for example, when it's an oddball. Yellow just means an increase in that particular power spectral band. When we take the difference between those two, we get this difference plot over here. And this difference plot is now saying how much enhanced power is there if it's yellow when you look at the oddball versus the non-oddball stimulus. And if things are blue, that means the opposite. There's more power when the stimulus, in this case the AAAA, is fully predicted. And so what we found there is, again, the spectral signature of enhanced processing to the unpredicted stimulus. Again, even when it's controlled for adaptation, this increased gamma also increases in the lower frequencies, the theta range, but decreases in the alpha beta range. So in addition to that, my prior work with when I was at MIT with Earl Miller showed that this enhanced gamma response to an unpredicted stimulus, but reduced beta response, was layer specific. And now that's going to turn out to be, I think, very important because the exact layer of processing is going to determine which types of cells are getting engaged in this computation. And not only that, where are these cells then projecting? Are they projecting feed forward, which is happens in the superficial layers, layers 2 and 3 of cortex? Or is it projecting in the feedback direction? And that's the layers 5 and 6, these deeper layers of cortex. And so when we look at unpredicted versus predicted, how much signal change is there, what we found is that this was really much stronger in superficial layers for the gamma. Not only that, it was also stronger in the columns that represented the predicted stimulus. So it's not that gamma is going up everywhere in the brain. No, it's the fact that gamma is going up really at the next to the neurons that actually encode the predicted features. So the predictions do reach down at the specific level that is necessary and to the specific neurons that are necessary to get enhanced. And the same thing was true for beta inhibition, but the other way around, right? So this negative sign means that beta is stronger again in the predicted case. But notice that the decrease in beta was only found in the columns that preferred the predicted stimulus. For the columns that did not prefer the predicted stimulus, there was no change in beta. And this is what we call predictive routing. This is the basic observation for what we think is a necessary update to the predictive coding model into a predictive routing model. So what is predictive routing? It's a neurophysiological implementation that's a little bit more realistic and true to the neurophysiology of predictive coding. And here's what predictive routing says: that when the prediction, which I'm diagramming here on the right, when the prediction matches the stimulus, so for example, we predict that we're going to see stimulus A, and we in fact get stimulus A. So this is the feed forward stream here. This is the feedback stream here. When they match, let's see what happens. When they match, these higher order circuits can synchronize other areas at alpha beta, but they synchronize where they're needed, only to the neurons that prefer that predicted stimulus. That's why we are depicting here neurons that are selected for the stimulus that is stimulus A, the predicted stimulus, and the stimulus that in fact arrives. So this top-down signal, it originates in a layer-specific way and targets deep layers. And it enhances the alpha beta at those locations that are necessary. So it prepares cortex. Predictive routing is all about issuing predictions. And what do the predictions do? They prepare you to receive the sensory stimulus that is predicted. And what happens when there's a lot of alpha beta in those circuits? What happens is that lots of other work has shown that this alpha beta has an inhibitory effect on the superficial layer gamma and superficial layer spiking. So as a result of enhancing something that's inhibitory, you decrease the activity of the gamma and the spiking. And so as a result, that's why this wave here is relatively low in amplitude. There's relatively little feed that feeds forward. You don't have to encode that sensory information. It's been predicted. But what happens when you do not successfully predict something? Well, all of those mechanisms that I just highlighted are not present anymore, because now you've predicted the wrong stimulus. We've predicted B, but stimulus A has arrived. And so as a result, higher order cortex has not prepared these A processing neurons. Those A processing neurons are in a relatively disinhibited state, a state of lack of preparation. And in this lack of preparation state, with reductions in alpha beta, this cortex is going to be more disinhibited, and therefore stimulus A, when it comes in, it's going to get processed much more vigorously and evoke larger gamma amplitude waves and larger spiking activity and feed forward more into the system. Okay, so that's our model. And now, how is this related to consciousness? So we tested this model. We're testing this model in many different ways. One of the ways that we're testing it is to actually record from subjects as they're experiencing predictable or unpredictable sensory stimuli, as we just showed you over the last few slides, but doing it both in the awake state, where these predictive routing mechanisms are fully intact, as well as in the unconscious state. In this case, we're going to use propofol in order to very rigorously control the state of consciousness. So we're going to induce unconsciousness using propofol, and then we're going to see what that does to this predictive routing. Okay, so here's our paradigm. We're using, again, a very similar paradigm where you have both locally repeated stimuli as well as occasional local oddballs, as well as a sequence. In other cases, you can create a sequence level prediction. So beep, 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 boop, beep, 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 boop, and then you can create a locally predicted stimulus that is a global oddball. So that's these trials right here. And in this paradigm, we're going to look at both of those types of stimuli, both the local oddballs as well as the global oddballs. Our propofol administration strategy was to record from this paradigm. This was an auditory paradigm in this case. The animals were just simply in a resting state. They were played these tones in the awake state. Then we induced loss of consciousness. We maintained them in a unconscious state. This unconscious state was characterized by the closing of the eyes, several other peripherally measured signs that are consistent with loss of consciousness, as well as the presence of slow wave activity and the reduction overall of spiking activity and the reduction overall of faster frequencies. We did not induce a profoundly inhibited state of cortex. So we did not, for example, have electrotonic periods of silence. In fact, this was a slow wave regime that we implemented. So, and we titrated that to achieve that particular cortical state. Now, during that cortical state, as well as during the awake state, we recorded in two distinct areas, okay? A higher order cortex area. So this is the macaque brain. This is the front of the macaque brain. So this higher order cortex area is area FEF. That's in prefrontal cortex. Again, this is the area that we think issues the predictions and issues the control of the sensory cortex through these alpha-beta oscillations. And then we also recorded from a sensory area, in this case, part of the sensory stream in the auditory sensory stream, area TPT. Okay, it's also part of the superior temporal gyrus. It does have a very strong homologue to the human superior temporal gyrus area, and this is an area of relatively higher order processing in the auditory stream. It does also get, interestingly, multisensory cues and multisensory information, but it does have very strong responses to the auditory cortex or auditory processing. Interestingly enough, so does area FEF here in the frontal cortex, and it's been shown that these areas are monosynaptically connected, okay, both in the feed-forward direction as well as in the feedback direction. So let's see what happened to these neural responses. We're going to start by plotting here the spiking activity, okay? So this is the individual neuron response. In this case, again, I'm averaging the activity of many individual neurons. In this case, we're recording between 100 to 200, depending on the context and area here. So this is a large amount of data to give us that view. Once again, just when we look at the average amount of activity happening to each area, what's going on? Where are these things getting encoded? So that's the first question that I'm showing here in this slide. And again, on the x-axis, I have time. So we have different oddballs or non-oddballs happening in red, different auditory inputs. The black is when there is a local regular, so that's just a locally repeated stimulus. The blue is when there's that release from adaptation, that what we call that local oddball, that beep, 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 boop. So that beep, 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 boop signal in the lower order sensory cortex is very robustly encoded and very early in the sensory response. Now, in the higher order cortex, in the awake state, that's this left column, there is still, of course, a very enhanced and very robust detection of that local oddball. It is interesting that the amount of adaptation is very profound in the higher order cortex. So auditory cortex, to each repetition, does adapt, but this adaptation occurs after just one single tone. It's almost like higher order cortex is saying, okay, all I got to encode is the beginning of the tone sequence, and then I know everything that's happening next, unless there's an oddball, then I got to encode that as well. So already just at this very low level sensory processing, we already see really interesting evidence here for differences in adaptation and perhaps differences in the computations that are being performed. But now let's look at what happens during propofol. Okay, so that's the right column. So what happens during propofol is that, first of all, and somewhat surprisingly to me at least initially, the low-level sensory areas are still quite strongly encoding those sensory stimuli, independent of their predicted or unpredicted effects, right? So it's not like the brain is completely turning off. And in fact, the sensory cortex response, at least relative to the baseline, is still quite profound and quite robust. Now, notice what happens though when we get to the oddball, right, which is right over here. What we see is that the amount of oddball processing that we find is similar in nature, at least early on, and then occurs for a longer period of time. So what we call what we interpret this is to say that there is a disinhibition, in fact, of sensory cortex when you look at the unpredicted versus the predicted signal. Okay, so this is already consistent with the fact with a loss of frontal inputs. Okay, so with the loss of these higher order control signals, which we think are inhibitory through alpha beta, we see that there's an enhanced oddball response. Now notice what happens in FEF. In FEF, there's a much more profound reduction in activity, and in fact, there is actually no effect of the oddball anymore. So that's again this observation that frontal cortex is very profoundly inhibited during propofol mediated unconsciousness here with propofol, and that this lack of frontal inputs then might in fact leave sensory cortex in a disinhibited state. So in addition to these local oddballs, we also wanted to look at whether or not there were responses to global oddballs, right? So following up from the first data that I showed. And what we found is that in this paradigm, there was global oddball, but it was at the level of decoding. So what we did here is we looked at across many different spiking neurons and said, whether or not that population of activity can give you decoding of whether or not the same stimulus, AAAA, for example, was a oddball stimulus, an unpredicted stimulus, by comparing whether or not those neurons had information on whether or not it was a predicted stimulus compared to the same stimulus, AAAA, when it was a fully predicted stimulus. So what we found here, and I apologize, the red and the blue aren't labeled here. The red is anesthesia decoding. The blue is awake decoding. And so what we found is that the sensory cortex, it does not decode whether or not a stimulus is a global oddball. Of course, it does decode what the stimulus was, and it does decode whether it's a local oddball. But again, when we control for this sensory adaptation, there's no decoding, neither in the awake state nor in the unconscious state in area TPT. But what we did find is that in higher order cortex, FEF, as well as prefrontal cortex, we found that there is significant, albeit a little bit on the lower amount, but this is this line here is 50%. It's chance level decoding. So anything above the line is statistically significant decoding of the global oddball. And we found that there was significant awake level decoding of global oddballs, but that this fell to chance levels in the state of propofol mediated unconsciousness. So we do think that global oddballs are an emergent property of higher order cortex and that they depend on the wakefulness state. In addition, what we did here with this analysis is we wanted to look at not just these global populations of activity, but we wanted to get a sense for the individual neurons, okay? What are these individual neurons doing? So each point on these scatter plots here now is an individual neuron that we recorded across our sample. And now these individual neurons, we're plotting them for on the x-axis for how much of a local oddball effect did each of those individual units have. So how much did they respond to beep, 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 boop versus beep, 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 beep? On the y-axis is how much did that individual neuron respond to the global oddball effect? And then the neurons that are labeled in black are the neurons that had significant encoding both of the local oddball as well as the global oddball. So what we can see here is that in both areas, there is a little bit of global oddball effect. Interestingly, though, again, in area TPT, this global oddball effect was not sufficient in order for us to be able to decode the global oddballs, but it was sufficient in area FEF. And then a lot of channels, of course, do not really have robust responses to neither the local oddball effect nor to the global oddball effect. So they're not really participating in this task. And those are the neurons shown here in gray. Now, again, this is the awake state on the left. Now let's see what happens to these populations of neurons when we move from the awake state to the anesthetized state, again, with mediated by propofol. Well, what we found is that in the anesthetized state, these vector lines are showing for each of these populations, these 4 populations, what happens to their neural activity on average, where do they move? And in general, in the anesthetized state, they move on this global oddball axis downward. So they don't change as much on the local oddball axis, but they move downward. So in other words, that global oddball response, that higher order type of response, is reduced. And in the higher order cortex, this response is reduced 100%. Everything moves towards this boundary of insignificance. So nothing is activated. It's really profoundly inhibited. But notice one very interesting effect, and this is again evidence for disinhibition. What we're seeing is that before we had lots of channels that were not interested in the task in the awake state, but in the anesthetized state, this arrow right here is showing that, and these gray dots are escaping the boundary of this insignificance. So again, there is evidence here for not only just overall reductions in activity, but especially for neurons that weren't previously participating in the task. Again, perhaps in the absence of higher order signals, we're seeing that lots of those neurons are disinhibited, are escaping from this insignificance boundary and now become activated mostly by the local oddballs. Now, again, I told you about how in predictive routing, we think that what higher order cortex is doing is issuing predictions at alpha beta that are then contributing to this inhibition, right? This control of stimuli when they are predicted. So now what is the evidence for that? And does that process of frontal control, especially at alpha beta, does that actually change? And we found that it does, okay? So here's how we did this analysis. We looked at the local field potential, the different brainwave oscillations, again, time locked to the onset of that oddball stimulus. This is the local oddball here. And again, what happens in just sensory area TPT in the awake state? Well, we've already seen this before. There's an enhanced gamma response. So yellow means an enhanced power to the unpredicted local oddball stimulus. There is, on the other hand, a reduced alpha beta response to that local oddball stimulus, or again, an enhanced alpha beta during that predictable stimulus. This happens strongest in area TPT, although it's also evident in area FEF. But now what happens when we anesthetize and give this same local oddball is that the alpha beta modulation is completely wiped out in both areas. And instead, what happens to the gamma is that it shifts down in frequency and it gets extended in its response and it enhances its amplitude of response as well. Again, FEF is almost completely out of this picture. Higher order inputs are out of the picture. What happens when you then release that top-down inhibition? Well, sensory cortex is in this uncontrolled, enhanced gamma state without the ability for the top-down beta to bring it back down and control its state. Now, you might think, okay, well, if sensory cortex is disinhibited, it's got enhanced gamma, it's got enhanced spiking when you look at unpredicted versus predicted, why is it not then capable of driving the higher order cortex, right? So what we found is that despite all of these effects of disinhibition in sensory cortex, those neural responses just don't make it out. They are not capable of driving the higher order FEF neurons. And the reason that we think they're not able to do so is because the the spike field coherence, okay, so how much the spikes in one area are coupled to the ongoing brainwave oscillations in other areas, that is in fact also profoundly reduced. So what we did for this analysis is we computed the spike field coherence between spikes in the sensory area, TPT, and the higher order cortex, FEF, at gamma frequency. And what we found there is that that degree of coupling, which is shown here on these upper graphs, that degree of coupling is profoundly reduced when you go from the awake state to the anesthetized state. And it's profoundly reduced, interestingly, in both directions, both in the TPT spiking to FEF gamma, as well as the FEF spiking to TPT gamma. So there's this profound reduction here in this long-range gamma oscillatory coupling. Not only is there a profound reduction in the amount of coupling, but the phase at which the spikes couple to the gamma is profoundly shifted, and that might impede the neurons in effectively communicating. So we think by a combination of both of these factors, there might be profound reductions in how the sensory areas and the prefrontal areas are communicating. So again, despite the fact that there's perhaps a disinhibition effect, those effects due to the lack of this coupling, this long range interareal coupling, there's no more the higher order cortex just isn't capable of listening to what's happening. So I'm already going to go ahead now and conclude the talk. What we've seen here is that these Madeleine recordings reveal that the local oddballs emerge early. But those local oddballs, they conflate prediction with adaptation. When we isolate predictions, we find that they happen much later in the processing chain than we had hypothesized. And we find that there is this control over the predictive coding process by gamma beta oscillations, which we propose as the predictive routing model. And furthermore, what does this have to do with consciousness? What we found was that propofol destabilizes this circuit. So it does so in two ways. There's sensory disinhibition, but in the absence of gamma beta interareal communication, that sensory disinhibition doesn't make it out. And so therefore, those higher order networks, what the global neuronal workspace might refer to as ignition, that ignition of higher order cortical recurrent circuits doesn't happen, possibly due to this lack of oscillatory drive. And so with that, I know I'm a little over, so I'm gonna skip that last section. And thank you for your attention, and thank you to my collaborators at Vanderbilt and at MIT, to my financial support, and in case you have further questions, please reach out. Thank you.
Andre, that was excellent, very clear and wonderful work. I really love this paper when it came out. We have some questions. I'm going to take the prerogative of one question that I've been wondering since I read the paper. Under propofol, the gamma is not abolished. In fact, it can be enhanced. Is that right?
That's right, yeah.
So that would go against the idea that anesthesia acts by inhibiting GABA and whatever receptors mediate that.
It does indicate that there is some amount of residual processing, but it could be through GABA B receptors, even though they have maybe very slow time constants. One of the things that we have found in computational modeling work is that you actually do not need exclusively fast timescales in order to get gamma. What you need is a particular coupling pattern between neurons, and that is sufficient to give you to give you the gamma. And so again, of course, propofol is a GABA A agonist, right? So it doesn't have as much effects on all forms of this inhibition that is necessary to drive gamma.
Great, thank you very much. Let's start over there.
Yes, hi. Thanks so much for a great talk. Andrew Budson here, Boston University and the Boston VA. There's a lot of debate in the consciousness field about whether the back of the brain or the front of the brain are the areas that are critical for consciousness. And I was thinking in your talk that given the fact that the back of the brain, the TPT areas are still able to process information, but the front of the brain, the FEF, are not with propofol anesthesia. Is it correct to assume that your research would suggest it's actually the information reaching the frontal eye fields, that's critical for conscious perception?
Yeah, I think the data thank you for that question. I think the data are actually very strong on this point. Our data do in fact suggest that the posterior cortex is intact, is relatively intact, not fully intact, but relatively intact, as you can see here on the slide, during states of loss of consciousness. But the front of the brain is more profoundly affected, and of course also the interactions between frontal and sensory areas are very affected as well. So yes, I do believe that this data is speaking more to the, if we want to be a little simplify things, the front of the brain type hypothesis for consciousness, or at least the front of the brain to back of the brain and vice versa interaction and communication structure for consciousness.
Thank you.
Please be brief.
Thank you for the talk. I remember that in the dendritic integration theory by Matthew Larkham says that the superficial area is rather feedback, and the deep areas are cortical columns are more feed forward, provided by thalamic input. So I think there is a little bit discrepancy with your idea and theory. So I just wanted to clarify how I can understand that.
Yes, it has to do with my apologies. I was slightly sloppy with my terminology. So superficial layers, what I refer to as superficial layers is layer 3, which house the superficial pyramidal neurons, and those neurons do feed forward. Now, what Matthew Larkham is discussing is the deep layer 5 pyramidal neurons that have their apical dendrites that reach all the way up to layers 1 and 2. And so these layer 1 and 2 layers are receiving feedback, okay? So the very highest, most superficial parts of the cortex, those are in fact feedback recipient layers, and those were not the layers that I was referring to when I was referring to superficial. I was referring to the superficial layer 3 specifically, part of that feed-forward processing circuit.
Thank you.
Thank you for that clarification.
I'm very sorry, but we got to cut off the 2 questions. I apologize to both you guys. Let's give Andre a round of applause. That was a great talk.
Thank you so much. I really appreciate it. And if you do want to follow up on any questions there, just please reach out.
Okay. Oddballs are going to become very, very popular.
Thank you.
Okay.
Thank you.
Our next speaker, it's a pleasure to introduce PULIN Gong from the University of Sydney, and he will be speaking on spatiotemporal activity patterns in the brain, dynamical properties, and functional roles. PULIN Gong.
Thank you. Thank you, Stuart. Thanks for inviting me. And it's my pleasure to be here to present our research findings to you. So today I'm going to talk about spatial activity patterns in the brain, about their properties and also their functions in cognition. So as we know that cortical circuits are composed of many, many neurons, with 80% being excitatory and 20% being inhibitory. So those neurons, they work together to give rise to some sort of complex, rich activity dynamics. And well-established finding is that when you look at this figure here, we can see that's spikes of individual neurons. You see spikes are very, very random, very, very variable, okay? That's just for one neuron for different trials. After stimulus onset, of course, fun rate would go up, but still spikes are very, very variable. However, at the circuit level, we can see that some sort of coherent structures or spatial temporal patterns can emerge. So this is the finding from my group. So we've done this experiment with our collaborators. So that's Utah array recording for MT area of marmoset. This is for anesthetized animal. We can see these slow oscillations. Because we have slow oscillations around 2 Hertz, and then we can extract phase of slow oscillations. And based on phase, and then we can come up with that phase map as shown there. But looking at this phase map, you can see this sort of rotational structure, okay? Some sort of patterns there, indicating that activities at circuit level are not totally random. There's hidden order there. So this emergence of order or patterns or coherent structures from otherwise random variable spikes. So this scenario is very similar to the emergence of coherent structures or vortices from turbulence, right? Turbulence is very complex behavior, right? So in turbulence, to characterize those coherent structures, people often introduce some vector fields or velocity vector fields. By looking at velocity vector fields, you'll see some singularity points or critical points. Around those critical points, velocity is around 0, right? So like saddle point and a source point whatsoever. So as soon as you know those critical points, you will know the overall spatial temporal dynamics of your turbulence, okay? So that's the idea of our methods. So by adapting some methods from the field of turbulence, we've developed a methodological framework to characterize spatial temporal patterns in the brain. So look at that, those figures. So by introducing vector fields and seeing turbulence, we found all kinds of interesting patterns. The first row, that was source pattern. You can see activity propagating toward a singularity point. And also sink pattern. We can see activity propagating away from a singularity point, as well as spirals, right? So I'll talk about spirals later. And also saddle point, okay? This kind of interesting spatial temporal patterns. So those patterns, they're not, I mean, as I said, they would play very, very important functional roles in our cognition and perception. That's something I'm going to talk about shortly. So we applied our methods to slow fluctuations of fMRI signals, because we know that, okay, for fMRI signals, we have this called very slow fluctuations as shown there. So you, for those slow fluctuations, right, which are around 0.01 hertz to 0.1 hertz, very slow fluctuations or oscillations. For those slow oscillations, we can extract, as I said, phase of those oscillations. And then we can obtain phase map as demonstrated there, and then we can introduce vector fields as what I've just mentioned. Based on vector fields, you can see these kind of beautiful spatial temporal structures or coherent structures. As demonstrated there in panel C, we can see rotational wave patterns or spirals or vortices in fMRI signals of the human brain. So red color indicates that that vortex or that spiral would rotate in anti-clockwise way. Blue color indicates that that spiral would rotate in a clockwise way. By looking at the panel C, panel D, you will see that those spirals, they tend to be located near the boundaries between different brain regions, okay? So why those spirals, they tend to be located around boundaries? That's just because as compared with the plane wave, you have plane wave like that, right? Rotational wave or spirals, spiral waves would have extra degree of freedom, which is rotation, right? Because of this rotation, so spiral waves can coordinate activity to flow out from our area and then into another area. So spiral waves or vortices can work as sort of mechanism to coordinate large-scale flow of information in the brain. So that's the idea. And so I'm going to elaborate on this later. And also we found that as shown in panel E, you see amplitude, okay? If we move away from a singularity point of a vertex, amplitude actually increases. So this is indication about the presence of spirals or vortices as found in other complex systems such as turbulence. So here I will show you a movie. Okay, so you see the many spirals, okay, moving around, interacting with each other in very complicated way. Okay, so that's this kind of rich and complex spatial temporal dynamics I was talking about. So as I said, spirals, they have this kind of rotational motion. this rotational motion is very non-stationary. So sometimes they rotate very quickly, sometimes they rotate very slowly. So this is very dynamical, non-stationary rotational motion. And that figure, as demonstrated there, okay, we have the trajectories, okay, trajectories of many spirals. So the immediate impression you can get is that those trajectories are very variable as well. Just like real oscillations in the brain, real oscillations in the brain are very variable. So the real propagating waves in terms of their dynamics should be very variable as well. If you see regular propagation waves, so that could be troubling because that may correspond to some sort of epileptic state. So these variable trajectory propagating dynamics actually can be mathematically characterized as super diffusion, okay, as demonstrated in that figure. It means that it propagates faster than normal Brownian motion, okay, random motion. And it's not totally regular. It means that there's a mixture of ordered and disordered behavior for the propagating dynamics of vortices. As I said, okay, they interact with each other. And so here, when you see, we have for the panel B, you can see anti-clockwise spiral, and then it would interact with a clockwise spiral. So during this collision process, actually, spirals can reorganize the overall spatial temporal dynamics. Before collision, you will see a propagating wave, plane wave, okay, from left to right. But after collision, both of them, they would disappear. They are dead. And after that, you see the overall dynamics would be organized in terms of a plane wave from right to left. Okay, so it means that interacting spirals can organize overall large-scale spatial temporal dynamics. So this is analogous to the function of vortices in turbulence, because vortices in turbulence can also play this sort of role for organizing overall spatial temporal dynamics. And of course, that just for a pair of vortices or spirals. As I demonstrate to you, we have many spirals, okay, that can interact with each other in a very complicated way, and particularly in this cascaded way. Okay, so one pair of spirals can interact, and then the outcome of that interaction can then interact with another spiral, so on and so forth, to coordinate spatial temporal dynamics of the brain. So those spirals, I mean, of course, they have some sort of functional correlates. So we look at the language task data from HCP, and we found that, okay, for specific tasks, okay, those spirals, okay, they have specific locations, and also they have specific rotational directions. In other words, so the rotational direction and location of spirals are task specific. So just give you an example for this language task. For story listening stage, you'll see this anti-clockwise spiral near PCC area. And, but for math listening task, it would be clockwise spiral, okay? Clockwise spiral. And very interestingly, those spirals, they are symmetrical across both hemispheres, okay? You see, that's because this way, you get rotating this way, all right? So this symmetrical property, that's why rotational directions are reversed for left and hemisphere vortices or spirals. And also this is for another stage of this task. So for story listening and story answering and math answering, again, we have this specific spirals with a specific rotational direction and specific location. It means that they are task specific. And also we can use very simple linear classifier, a decoder, to read out information from the rotational direction and the location of those spirals. Okay, so with very high accuracy, okay, so much higher the chance level as compared with the standard methods like functional connectivity or whatsoever. So that's for language task. So the similar thing can happen for a working memory task. All right, so that's for this fMRI data after filtering in specific spatial scale, as often done in standard functional connectivity analysis. We also look at unfiltered fMRI signals, and we found that those spirals are very robust, even for unfiltered data. So this result indicates that spirals are robust property. okay, robust property of the brain. And also, as soon as you have this rotational structure, right, you can project your activity onto low-dimensional components or manifold. You'll see this sort of rotational component because in some neurophysiological recordings, right, people often observed some sort of rotational manifolds. Okay, they thought that those rotational manifolds could correspond to some behavior tasks, right? But in our framework, those rotational manifolds are just this kind of byproduct of our rotational wave patterns. Those spirals, they interact, okay, as I said. So here, okay, you can see for the first figure, okay, we have clockwise spiral and anti-clockwise spiral. They interact. Because of this interaction, activity can be guided to flow through their interaction region, right? But if you have two spirals with the same rotational directions, okay, anti-clockwise, anti-clockwise spirals, you see activity would be prevented from flowing through the interaction region. So in other words, so interactions of spirals can be used to organize flow of activity. And based on this principle, actually, if you have multiple spirals working together, as demonstrated in that figure, we can have this kind of guided large-scale flow of activity. So we applied this organization principle to real tasks. Okay, so the figures are just showed to you. So those figures are, of course, based on real data. And then we applied this organization principle to the task data. So we found that, okay, so first we identified task-specific region, okay, and show in the top figure, those areas, okay, is a region of coordination. And then we look at different task conditions, math listening and math answering conditions. And as demonstrated there, around the bottom, around this here, okay, you'll see all these spirals, okay, working together to guide activity flowing from a bottom-up direction, okay, bottom-up direction for the initial stage of this task. But later on, for the math answering condition, as demonstrated there, because the rotational directions of those spirals would be reversed, so that the overall flow of activity, okay, the direction would be reversed as well. So now the activity would flow from top to down direction, okay? Initially, it's from bottom-up direction. So it means that, so this interaction spirals, okay, can organize this flexible flow of information. And we built a mathematical model showing that, so this mechanism, okay, can also happen in our mathematical model, okay, suggesting that, so these interactions play a very important role in organizing brain dynamics. And then here, I mean, there's a very recent paper. In this paper, people found similar multiple spirals, okay, in I think this is cancer imaging data from the whole cortex of mice. As you can see, there are multiple spirals there, and so you can see, particularly for right hemisphere, there's one here, another one there. They interact. Because of that interaction, activity can be guided to flow through their interaction region, as we've demonstrated in human brain. And also, as we found in mice brain, people found that the densities of spirals, okay, are around the boundaries of different brain regions, okay, as demonstrated in that figure. So consist with what we found from a human fMRI data, suggesting that so interacting spirals could be a general property for organizing large-scale brain dynamics. And also, we have a paper at the review at this stage. We look at sleep spindles. So this is for N2, stage 2, no rapid eye movement sleep. So for the N2 sleep, as you know, the feature is about that spindle oscillations around 11 to 16 hertz. So for these spindle oscillations, as demonstrated here, we can see these beautiful spiral waves rotating around the cortical surface. So the first time I saw this, I was shocked because, I mean, we didn't do any filtering process, right? And still, you can see this beautiful, regular, this kind of interesting, dynamical spiral waves Of course, spiral waves for spindle oscillations have been observed in ECOG signals, but for the first time, okay, we found evidence that the stability or the consistency of a spindle spiral wave actually can be used to predict memory performance. Okay, so these results suggest that spiral waves of spindle oscillations are crucial for memory consolidation. Okay, because the idea is that you have this big wave rotating across large part of your cortex, right, during sleep. So this can integrate different inputs like auditory inputs you receive during daytime, or sensory or visual inputs, okay, during daytime. So they can integrate multi-modality inputs, okay, you encountered during daytime, okay, so that memory can be consolidated. And also, very interestingly, we found that the consistency or stability of spirals of spindle oscillations, actually, the consistency decreases as we are getting older, okay, as we are getting older. So this suggests that spirals could be a good marker for aging, or even for some other brain disorders, such as dementia. All right, and also we found that, this another study, okay, our ongoing project, we found the spirals ubiquitous, and also in MEG data. So this is this kind of spiral waves, interacting spiral waves near motor cortex of the human brain. As you can see, there are many these sort of like spiral waves, as we've demonstrated based on other recording modalities. So interactions of spirals, okay, they're important. So many years ago, I proposed a theory in terms of computation, distributed dynamic computation based on interactions of wave packets. Okay, so you have one wave packet here, you have another there, right? Information could be encoded in wave packets, and the information could be propagated because of the propagation of wave packets from one area to another area. And then information could be processed while they interact with each other, right? So in this way, because you have many interacting wave packets, so the brain may do this distributed dynamical computation. So based on these transient interactions, instead of stable attractors or whatsoever, okay, if your brain is in stable attractor, you might be in big trouble. So here, in my way of thinking, the brain may use this sort of complex non-stationary dynamics for doing flexible, efficient, distributed dynamic computation based on interacting wave packets. Wave packets could be spirals, or could be source or sink, or other types of waves. And so this distributed dynamic computation actually is analogous to a very famous abstract mathematical model, computation model, which is called billiard ball model. Okay, billiard ball model proposed by Friston, yes, in 1982. So the idea is that you have two balls, right? They would have this elastic collision, and the computation can be done during this elastic collision process. So motivated by this, actually, Richard Feynman proposed quantum computation for the first time. The paper was published also in 1982. So this paper, okay, the billiard ball model, the idea is to link physics with computation, like mechanics, collision of two balls for doing computation, okay? So how physics can do computation. So as I said, I mean, because those wave packets or spirals, okay, they can move in this very complicated way, as demonstrated there. So this propagation can be mathematically formulated as super diffusive Levy motion, okay? So it means that your pattern, okay, your wave packet can stay here for a while, and then suddenly shift or jump to another location. So this is super diffusive Levy motion, okay? For example, when birds search for food, birds would fly in that way. So first, birds would search locally, and then suddenly, and then you have long-range flight, okay, to another location. So this is, and people, this is optimal. Actually, people prove this optimal searching behavior. And also for vortices, in vertex, vortices would move in this super diffusive Levy motion. Oh, just based on this, okay, this specific, this kind of complex rich dynamics, we've developed a theory for understanding how this rich spatial temporal dynamics can can do neural computation because, I mean, we have dynamics, but dynamics are not there as epiphenomena. They should play some sort of computation roles, okay, and then can lead to perception and cognition. So the idea is that the movements, this complex spatial temporal movement dynamics can be used for doing sampling-based probabilistic computation, okay? So the idea is that while the pattern is here, okay, your pattern can sample that information. While it moves there, it can sample the information there. Actually, so we know that many cognitive functions, okay, can be formulated as probabilistic inference, okay, Bayes inference framework. So the fundamental problem is that how probabilistic distributions can be represented and processed in the brain. Okay, this is a long-standing problem. So we just propose this theory for addressing that problem. Of course, there are some other proposals, okay, as demonstrated there in those scientific papers. So in those frameworks, Brownian motions, okay, like Brownian motions can be used for sampling probabilistic distributions. Because for Brownian motion, as compared with this complex Levy motion, Brownian motion doesn't have this long-range jump, okay? So that Brownian motion can only sample local probabilistic distribution, right? So, and then, I mean, to illustrate how this computation works, we first built a realistic spark neural circuit model, okay? And then we mathematically formulate this computational mechanism, okay, from algorithm level. Think about David Myers' 3 level of understanding of the brain. But anyway, so this circuit model is realistic. It's balanced, just like real brain. We have excitatory inputs and inhibitory inputs to individual neurons. They are balanced, okay? And also we have many other realistic features, and also it has adaptation, neural adaptation. So the working regime of our model, as demonstrated there, if I ratio, okay, look at panel A, if I ratio is high, so we would have this desynchronized state, which is classical state. Every spikes, okay, are not correlated, okay, they are in this asynchronous state. If we increase excitation, we would get global, I mean, as shown in panel D, global traveling wave, okay? So that's not good because, I mean, your brain might be in this kind of like epileptic state. The right working regime for our theory or for our model is near the boundary, near the transition stage between asynchronous stage and this global, regular propagating wave stage. So in physics, they say that somehow your brain is near transition between different states, okay, critical states. And in this stage, we have these kind of local patterns, moving, jumping, behaving in this super diffusive Levy motion way, okay? As I said, okay, this anomalous super diffusion can be used for performing sampling-based probabilistic computation, okay? And so that's a generative model for our theory, and we have feature space, I mean 2D feature space as demonstrated there. So when the pattern is there, okay, your wave packet can sample the posterior distribution and that feature space, during this sampling process, the dynamics of our wave package, as demonstrated there, still follow this super diffuse Levy motion. Okay, you have local movement, long-range jump, local movement, long-range jump, and very importantly, during this sampling process, okay, the pattern also exhibits theta oscillations. Okay, as demonstrated there, theta oscillations are extremely important for attention, for working memory, for all cognitive functions, almost. Okay, and also we demonstrate indeed, as predicted by our theory, so those patterns can do sampling-based computation. And so based on that circuit implementation, and then we have this kind of abstract mathematical theory, and so based on this theory, we know that our sampler, okay, wave packets can sample any complex probabilistic distributions. Particularly if you have two distributions with far away modes, right? You have one mode, another mode. If they are far from each other based on conventional theories, like Brownian motion-based sampling, because they are far from each other, your sampler would be trapped to local local mode, okay, without ability to switch to another mode. But that's not the problem in our framework because of this long-range jump. So our wave packets can sample here and then switch to another location to sample at another location and so on. And yeah, we can also see this in our circuit implementation. And based on this kind of theory, we can explain many experimental observations. So this really makes me excited, right? Because your model, your theory, not target to a specific experimental observations, okay, to quite many experimental observations. For this one, so this is classical motion detection task, okay, as observed in that paper, in that study. So the key feature is that there are bimodal distribution for that inferred posterior. One is around the prior, another is around the likelihood. So this is a little bit puzzling based on conventional Bayes inference theory, because for conventional Bayes theory, you have prior multiplied by likelihood. So you have unimodal distribution. So here, you look at, you have multiple peaks, right? But that can be explained because of our this anonymous super diffuse motion of our wave packets, because wave packet can stay here for a while and then to sample another mode. So we can explain many experimental observations. I'll skip the details of our explanation. And another very interesting point, I think, which could be related to the topic of this conference is that this computation based on this complex dynamics of wave packets can explain how attention works, okay? So attention actually is very flexible, very dynamical phenomena, okay? When I ask you to focus on two things, your attention may switch back and forth between these two objects. But occasionally, your attention may jump to something else, right? If your attention only focuses on these two things, okay, I think you're going to be in big trouble. But anyway, so we have this this new mechanism, okay, based on this complex dynamics wave packets for explaining how attention works. So in this framework, you can think about our wave packet as dynamical attention spotlight, okay, spotlight. So here, just like in real experiments, we have two objects, okay, we have two objects. So subjects were asked to focus on these two objects and then detect change of one of those objects. So we do similar thing in our model, and we have two objects, and then what we found is that so the wave packet actually can stay around one object for a while and then switch to another object, just like how our attention works. So during this process, when our attention pattern wave packet is focused on one spot, you will get burst. Okay, this kind of burst-like activity, while the switch to another location or less spikes around that group of neurons. So you can see this sort of bursting activity, okay, as found in real neurophysiological recordings. And very importantly, you see we have these 4 bursts per second. It means that we have theta oscillations, and as soon as we have bursts, the bursts would give rise to gamma bursts. Okay, it means that theta oscillations are coupled to gamma bursts, as observed in real experiments. You see, so that's panel G is figure showing how theta oscillations gamma bursts are carried by theta oscillations. Because theta oscillations, okay, if you look at like from wave aspect, theta oscillations, they are bigger waves, okay, they would carry local gamma bursts to do distributed dynamic computation. So that's about temporal aspect. Actually, from a spatial aspect, I mean, as I've just mentioned, okay, so the attention spotlight or wave packet would follow this Levy motion, as we've characterized based on these mathematical methods. It means that your attention will stay somewhere for a while and then jump to another location. So this actually would have profound computational advantage because it will address the long-standing problem about the balance between exploitation and exploration. Okay, exploitation in the sense that your attention will focus on what you want to see for a while, right? And then because of this intrinsic complex spatial temporal dynamics, occasionally your attention will focus on something else. So it means that these dynamics can provide you with a balanced exploitation and exploration. And also, as demonstrated in our paper, we found that this sampling mechanism is particularly good for sampling real nature scenes. Okay, it's not about simple two inputs. Okay, it's very good for sampling complex nature scenes. Okay, something we are dealing with every day. Anyway, I will stop here and thank you for your attention. I would acknowledge the contributions from my students, my postdocs, and also my collaborators. Thank you very much.
All right, a couple questions. Over there.
This is really great. I want to make sure I understand it. So yesterday we saw in, I think it was the Earl Miller talk, it was more grand kind of spiraling of these fields, right? And this is a more granular view of these little fields of firing, correct?
Yeah, yeah.
So, and you're saying that these firings, the waves can hand off this information is the theory, right?
Yeah,
so
yeah.
in essence, there's engagement of firing and information, right?
Yeah, yeah.
And then these will reach out to larger networks and then hopefully engage them or disengage them.
Exactly,
based upon.
exactly.
Fantastic. So it is very similar to nature and the way that we see creation in nature with spiraling in certain plants and these, and also weather patterns.
You're right, yeah.
So it's very similar to. So it's very similar to how pollen might spread throughout a natural system.
Yeah, yeah.
Correct?
Yeah, correct. Because I mean, as I said, because of rotational motion, information would be mixed, right? I mean, as compared, I'm used right. The rotation can mix information together.
Have you? I'll have to talk to you after. 'Cause I wanna take
Okay,
a-
all right, yeah.
Thank you.
Yeah.
A question from here, please.
Oh, yeah.
Did you consider on the basic level on physical-mathematical model, did you consider complex exponent as a solution for harmonic oscillator that is actually a spiral, geometrically? Did you consider the origin of your spirals? Where could they come from, mathematically?
Oh, yeah, yeah, we have mathematical model. We have ongoing project. I mean, that project, we are trying to build a large-scale model constrained by data. And then, so our preliminary result is that, so those spirals would emerge because of symmetry breaking. Okay, because of symmetry breaking, it means that some local patterns would lose stability. After losing stability, spirals would emerge. And they would have this kind of interacting and complex dynamics. So that we have a paper we are preparing at this stage.
That's great, thank you. And did you consider fractal dimensions? Did you determine fractal dimensions for your fractals that you consider?
Not yet. But I suspect the whole thing is not a unifractal, it should be multifractal because of heterogeneity in your system.
Sure. Thank you very much.
I asked you in an email whether
yeah,
these
yeah.
were coming from the apical dendrites or the pyramidal cells
Yeah.
causing the spirals. Any comment on that?
Yeah, I mean, because I mean, this is high level, this kind of macroscopic behavior, right? I mean,
Well,
I'm not quite, yeah, maybe it could be related because as far as we know that oscillations, okay, with different frequencies, they are coupled, okay? So slow oscillations would carry fast oscillations and so on and so forth. That may go down to the dendrite
I was
level,
thinking more of the mixed polarity microtubules going this way and that way in the dendrites.
Yeah,
that might
yeah.
oscillate back and forth. Anyway,
Yeah, we can talk about that later, yeah.
Thank you. I was hoping you could say,
over there to the right.
shall I keep going? So I was hoping you could say something more about cause and effect. The language you've used with regard to these spirals that you've recorded in the bold fMRI signal is that they are, I think you said that they are organizing the flow of neural activity, suggesting that they're causal. But is it equally possible that this is just some sort of structure that we see as an epiphenomenon of some other deeper organization of neural activity?
Yeah, I think that's very good, fair point. And so we are working on showing the causality here, but doing TMS study with my colleagues at Fudan University. But at this stage, everything is about correlation. Okay, just like the way how people do this sort of research for neuroimaging data, like functional connectivity. You have different mode network, different mode network would be correlated to something, right? So, but we want to go a little bit deeper to show this causal relationship as you just suggested. Yeah, we have an ongoing project on that, but doing TMS perturbation along with either MEG or fMRI recordings.
Thank you.
Okay, let's, the primordial consciousness man from India has had his hand up over here. So let's have him and then one more question over there.
Yeah.
Please, thank you. But please be brief.
There is a statement that the activities of one human brain are continuously influenced by the activities of the brains around him. Do you accept the statement?
Yeah.
Okay, then how far you are responsible for the activities of your brain?
All right. Yeah, I think I think I agree with you. So our perception cognition is emergent property because of interactions of our brain and the surrounding environment. Okay, if I put you in an isolated room without any sound, any sensory inputs, I'm quite sure you can't have any kind of cognitive function emerging in your brain. I agree. But the way how we can characterize these kind of interactions between different brains, I don't know. I think that's a very interesting question. Yeah.
Okay.
One more, please be brief.
Very brief. You mentioned the constancy of the spiral waves decreasing with age. Does the periodicity and rhythmic nature of spiral waves decrease with age, and have you considered also disease decreasing this?
Yeah, that's a very good question. So far, we only look at consistency, okay, in terms of a trajectory. Because the thing is that very amazingly, so as soon as we have spirals during sleep, they would follow the same trajectory, okay? And we found that this consistency of trajectory would decrease as we are getting older. But as I said, this could be a good biomarker, let's say, for brain disorders or something like that. That's something I will look at in the future. Yeah, very good
Thank
question.
you.
Great, thank you very much. Let's give PULIN a great round of applause. All right, it's my pleasure to introduce our next speaker, DIMITRIS PINOTSIS from the University of London, Brain-Computer Interface and Consciousness.
Thank you so much, Stuart. Can you hear me okay? Yep. Yeah, it's a pleasure to be here in this serene place and brainstorming with all these great ideas. And in a sense, I'll have a little bit less data than what we saw in the previous talks in my talk. And I'll try to speak a little bit at a more abstract level, at theory level, and interface with some of the ideas that many of you might be interested in with regard to consciousness. I don't do myself consciousness research. I have a lab in London, a methods lab, and the research directions that we work on are two. The first is developing new tools for brain scans, tools, for example, like the multi-electrode arrays and the laminar thin electrodes that we saw in earlier talks, not the tools themselves, but ways to analyze and explain the data from these new technologies, so statistical and biophysical ways and neural network models, as we will see. That's the first direction. The other is using these new technologies to develop and test brain theories, like predictive coding that we heard earlier, and we test these theories, again, using statistical analysis and machine learning, biology and math, in control populations and also in patients. So the focus of this presentation will be to talk about, a small personal, let's say, account of how I think one could develop a theory for brain-computer interface. And this is a hot topic, as we know, and it's in the news often because of Neuralink and other companies that have invested great amounts of money and lots of effort. What you see here is screenshots from a video that Neuralink uploaded a month ago with a patient. His name is Nolan Arbau. He's paralyzed below his shoulders after a diving accident. And for the first time, Neuralink said that using their device, this patient can play a game of chess. This, again, a video a month ago. And of course, brain-computer interfaces and brain implants is a big story, very hot at the moment, but long history behind it. You see a few examples here. In neuroscience, brain-computer interfaces have led to maybe the most important success stories, or among the most important success stories of neuroscience research recently. One by Helen Mayberg, that you see here, who has pioneered treatment for depression with using deep brain stimulation. And we'll talk about that later because we're working with Helen. Another success story is DBS for Parkinson. And of course, earlier, it goes back, the use of brain-computer interfaces to as early as 1748. And this British experimentalist that you see here on the top left, Benjamin Wilson, and took, of course, a couple of centuries, much longer, to have implantable cardiac devices. And the work by Eldmist and Senning in 1958 introduced the first cardiac pacemaker. The first cochlear implant was in 1961, not so far from here in LA by House and Doyle. So what does brain-computer interface do? Obviously, somehow it guides and interacts with neurons. You put a stimulation electrode near some neural populations. These are data that you see here by some beautiful work by Gene Friedman and his lab in the vestibular afferent. And on the vertical axis, you see changes in firing rate, measured somehow intracellularly, different sorts of neurons, excitatory and inhibitory. And then what you do is you bombard, you give pulses to those neurons, and of course how they respond and how the firing rate changes as a result of these pulses you see here in this diagram. And it depends, these peaks you get, these resonances, they depend on the intrinsic kinetics, the channel kinetics, the intrinsic physiology of different neurons. What are the challenges? For non-invasive BCI, of course, the challenges are multifold and relate to the low spatial resolution. Even with ultrasound, we still don't have a good understanding and control where the sources come from and how local the signal can be and how fast it can evolve, of course, and affect neural activity. Also, non-invasively, the main things that many companies, including Neuralink, focus on is avoiding the damage of brain tissue, body reactions, inflammation, the electrode, of course, degradation that comes with it. And I will say that the main challenge we have for BCI is really understanding what's going on and the biological complexity. So what are we stimulating and how the stimulation affects the biological properties, not only the neurons themselves, but also the cytoskeleton, including microtubules, filaments, organelles, everything that keeps neurons together. So I will argue that big data and good tech, even the best electrodes that don't cause damage, are not sufficient for getting good, efficient brain-computer interfaces, and there is a missing link that we are focusing on, and I guess others, which is biology, and theory based on biology and biological models, biophysical neural network models, midfield models like Amari, like Wilson-Cowan, like spiking models of the sort that Stephanie Jones and Nancy Kopell are doing, your favorite biophysical neural networks that describe the stimulation effects and the anatomy, the morphology, the physiology of neurons like axonal arbors, dendrites. Can you see my cursor? Maybe not. I don't know why. Do I have to move? Here it is. Okay, so the axonal arbors, the dendrites, the current flow, the neurotransmitters, the dendrites. We need to build models in silico implementations of cortical circuits and try to understand what's going on with stimulation. And the reason we need that, I will say, is because the reason we need biophysics is because interactions happen at the level of biophysics. The interactions between the stimulation, the brain-computer interfaces, the neurons that create this yellow aura around them, which is the intrinsic electric field, and also the cytoskeleton that has charged parts like the tubules and the filaments, and these have also contribution to the electric field. And then when we put a stimulation electrode, it interacts with these structures through their electric fields. Because, for example, what you'll see here is how microtubules are affected. Now, these are in vitro experiments, are affected by electric fields, as you will see here. So these have now random directions and orientations. And then they align. So, and then when this happens, so they change their structure as a result of the external field, then they go back and affect the dendrite configuration of the neurons. So there is this interplay between the microscale, the mesoscale, and back. So that's the thesis I wanted to present. And then I'll go one step further, and that will be my last, let's say, introductory point, that what many of you are doing, it's also useful, because not even biophysical models are sufficient, but we need also ideas from consciousness studies to understand how to build efficient BCI and theory for BCI. The reason for that second point is because the electric fields that are crucial, and their understanding that are crucial for describing interactions between BCI neurons and cytoskeleton, these same electric fields might be the actual substrate of consciousness. And there is a huge literature out there, the work of John Joe McFadden, Susan Pocket, Finkelkurz, etc., that suggests that. And there is a practical reason also that electric fields are very useful for controlling neural activity and guiding neural computation from the neuroscience perspective, because influences that they exert on the surrounding biological tissue happen at almost the speed of light and in distributed manner. Now, insights from the electromagnetic theory of consciousness that says that fields are substrates of consciousness also say that different conscious states will correspond to different configurations or special patterns of electric field in the brain. So that's the other intuition. So if we have a link between the biophysics, the biology of the electric fields, and the conscious states, we can see how the field we put with BCI can change the conscious state, and perhaps that could be a key step for mental disorders, and I'll come to that in the second application of the talk. But for the moment, I want to use this first insight that fields are substrates for consciousness, and different fields might thus correspond to different conscious states, to test experimentally, because we do neuroscience, the hypothesis one for this talk, which is that electric fields indeed might guide and influence neurons containing thoughts, containing different aspects or examples of conscious experience. And we have some papers with Earl Miller. You heard some of this from the excellent talk by Earl yesterday, some of this work, and also with Gene Friedman from Johns Hopkins, who is building this kind of devices. And I'll discuss that. So what is the evidence first for hypothesis one, that electric fields control neurons? There is a long history in neuroscience, something called the haptic coupling. And you see a classical study here, which says that mesoscale fields entrain spiking. So what you see in this plot from top to bottom is different external fields at different frequencies or periodicities. They go faster. And then you put this field on top of a slice where you have neurons. And then the spiking of the neurons follows the periodicity of the external field. And there are other also effects that extracellular fields might have, even intrinsic fields. The fields are not external imposed by the experimentalist, but the intrinsic fields like liquid junction potential, and there is a long history about that. Another study really fascinating that showed evidence that there is some kind of interaction between the mesoscale and the microscale at the level of fields and spiking is this study where by a group at Case Western University in Ohio, which shows basically, again in vitro, hippocampal periodic activity propagating, so it showed, a traveling wave on a slice from left to right, and then they cut the slice in the middle, and they take, I'm trying to find the cursor, yeah. They take the two pieces apart, panel C, and what do they see, surprisingly? They see non-synaptic interaction between the two parts of the slice, so a traveling wave that jumps the gap until the gap is 400 micrometers. If it gets bigger, of course, you don't. So what is this? It says that there is an ambient wave on the left slice that jumps and induces activity, and the traveling wave propagates through the gap to the right-hand side of the slide. So the question then in neuroscience is a haptic coupling a byproduct, an epiphenomenon, or does it have a functional role? And our answer, which is the answer to the first hypothesis that we want to test, is that the electric field is like the conductor in an orchestra of neurons, which guides and stabilizes them. So, and basically the musicians in the orchestra, not only neurons, but they include also parts of the cytoskeleton, hence the name cytoelectric coupling. And there is a coupling between the microscale, microtubules, filaments, proteins, and macroscopic oscillations, data networks. And this is really what also BCI would be doing, would be changing the conductor's behavior. And we have a couple of papers, like I said, these are the references with Earl and Jean that I have put on this slide. Okay, so what are results that show some support in vivo for this phenomenon? So if haptic coupling has been shown on slices in vitro, we have some results from in vivo data in non-human primates from Earl Miller's lab. So what we do is we use this biophysical neural field model, for those of you with a technical background, plus a model for the electric field. Again, I'm using the same word twice. The neural field and the electric field are separate. And using this biophysical neural network approach that allows us to reproduce in our computer what supposedly is happening to some approximation in the monkey brain, what do we find? We find, first of all, that the mesoscale electric fields contain information associated with our memory paradigm. So this is a special delayed saccade paradigm we are using here. The monkey needs to remember an angle, and then we have spiking data for that, and then we have the field data. And the field has also information. And basically what this graph shows here is the decoding accuracy on the vertical axis and the electrolocation on the horizontal axis. So for different locations on the patch, we can decode memory content, and importantly, the accuracy that we do it at the field level, at the conductor level, is higher than the accuracy you get when we decode spiking data. So as you might have guessed by now, to get this result, I'm using two biophysical models, one for spiking activity, so the neural model here. Let's see if I can, if I press here, yeah, maybe. So these are fluctuating waves, transient waves actually in this case, or standing waves that we predict from the cortex, from a small patch for the cortex that we record. And then on top of this spiking model, we have the electric field model that you see at the bottom. And just very simply, what is the electric field model is when you have the sources, the spiking, you can get basically the ambient field, the extracellular field around them. In the same way, when you use Newton's law of gravitation, you know the masses, that's sources, similar to the spikes, and then you get the electromagnetic field around. That's what we use to describe this interaction between the mesoscale and the microscale. What is another result here? So the second result, once we have these models for spiking and for electric fields, is to start looking for causality, right? If the hypothesis is that somehow the fields guide the neurons, because the fields correspond to consciousness, and somehow they are richer in information like we just saw, then if we use information theoretic measures, we should be able to find a directionality in these causal influences. And how do we do that? We use something called Granger causality, and with a little bit of twist. Granger causality says, look at the past of a variable A and a variable B, and assess whether your prediction for the next time point for variable B is better when you also look at the history and the past of variable A or not. If it is, it means that A influences B. But of course here we're talking about interactions using fields, electromagnetic fields, and these interactions happen at the speed of light, so there is no way we can look for basically history, because history means delays and the effect. So what we did is we used special Granger causality, so predictions over space and whether they improve. And indeed we found, basically looking at both directions, from field to activity, which is the left panel, the red bars, for different time points in our trial, where the monkey remembers a location, basically. Different areas here, FEF in the top left red panel. The bottom left red panel is SEF, another area implicated in the memory task. And then on the right panels, the blue, are activity to field. So what do we have here? We have the directionality from the field to spiking, and then spiking activity to field. And we see, you see that it is one order of magnitude larger, actually 2 orders of magnitude larger. So it's 0 to 10, and the other, the blue, is 0 to 0.8. And also it's more stable, and we have other arguments for that. So the coefficient of variation for Granger causality using the field to activity, the red, is very small variability, while the variability in the spiking in the activity to field direction is much larger, is 28 to 47%. So in the same way that the conductor makes very small moves and guides the musicians, the field, very little, but guides the neurons. That's a claim at least, or that's the interpretation of this result. What is another result? Another result is when we look at fields now between areas, right? The previous slide was just single area spikes to fields. That's what I saw here. Again, grades or causality. The left panel now has time on the vertical axis from the beginning to end, from top to bottom. And then these are different cued locations or angles that the monkey needs to remember. And now we look at grades of causality between the areas at the level of fields. And the yellow, basically, the yellow temporal windows that you see here, for example, this around 300 milliseconds after the, in the delay period while the monkey is remembering. When you see yellow, it means that there is a significant interaction at the field level between the field at FEF and the field at SEF. And this, of course, is not telepathy at the level of brain. What the interpretation is that there is synaptic propagation with some delays, of course, between FEF and SEF that brings neurons in the two areas together, like having two orchestras, let's say, one in FEF and the other in SEF, and they have to synchronize and play the same song. Here, the same song is remembering the same thing, the same angle. And of course, at certain points, when the conductors communicate, there is some synchrony also between the two areas, and this interaction is captured by this significant grazer causality. And this is known in neuroscience. It's called communication through coherence, that there are some windows of spiking activity that allow for transfer of information between areas. What we say here is that this transfer of information is more robust and is determined by the conductor, by the field, not the spikes themselves. That's the conclusion. And just for you to remember, maybe the cytoelectric coupling hypothesis, I have a video here that shows a completely distinct paradigm, basically a DJ with a speaker and the grains of sand, because this some sort of self-organization cannot happen only in neurons, right? It should happen also in biology. So let's play this. Hopefully. Yeah, somehow I cannot move it forward. Maybe if I go to that one. Yeah, we don't have sound maybe, but we have sound. Let's see. So what you see here, okay, I'll describe it because we don't have the music. So there is music playing, and then there is basically either fluid or grains of sand on the speaker. And then when the music changes, which is basically the field energy of the speaker, this leads to different patterns in the molecules of the water or the grain. And you see here the flow also changes. So let's go back. I think it's also shown. Yeah, unfortunately, I can't control the video easily here. Let's see, maybe here. Yeah, you see it also here. It has nice music, this video. But OK, you see now, for example, that he changes the music and the patterns change. Should be also, yeah. So he pours grains of sand on the speaker and then starts playing music. And you will see now that the patterns change. So these are different states of consciousness, if you will, to bridge it to your. And this, what we say is that the aggregate field generated by the neurons feeds back to them. Of course, this happens in an emergent fashion, and there is no DJ. But when you put BCI, that's what you do. You change basically the music, or you put a DJ there. So that's the wrap-up, how cytoelectric coupling might be working. And then I want to go to the second and last application, talk a little bit about preliminary data, where again, I'm trying to bridge biophysics, and consciousness and get inspiration from conscious studies. The main insight we had earlier, right, is that different configurations of electric fields, either intrinsic or intrinsic plus BCI, meaning the ones generated by neuron cytoskeleton or the ones we impose, correspond to different conscious states. So what is a conscious state, if you go now to philosophers of mind, right? How do they define it, or a state of consciousness? It has certain criteria that people talk about. One is sensory content. Basically, what am I sensing? What am I getting from my sensorium? And this could be also interoception, right? What is the signal that comes to my brain from my body? The other is cognitive content. What is the brain doing? Not what is getting as input, but what is it computing? Like, for example, executive control or attention. And then there are many dimensions that one can put about subjective experience. What defines a conscious agent? Could be the first-person perspective, the sense of presence, the quanta of time. And interestingly, all these references I put here is people linking electric fields to these subjective dimensions. So these are criteria that define consciousness states. And then let's say you want to build efficient BCI for depression, which is what we are trying to do with Helen Mayberg. Why should you even be bothering using all these tools? My argument is that, of course, depression has its own field because it is a state of consciousness. So it has its own sensory content. You have certain feelings of fatigue, interoceptive signals, tiredness, lethargy. You have cognitive content, of course. You have impairments primarily if you talk to psychiatrists with respect to executive control, agent's attention, experience of time. So all these criteria are fulfilled and define a distinct state of consciousness and electromagnetic field to the control state, to the non-patient state. So this is why perhaps working with fields also makes sense from the theory of consciousness and theory of mind perspective. So to wrap up, what is the assumption here? The assumption is that neurons and cytoskeleton produce electric field. BCI produces electric fields, and the two interact. And then what efficient, if you want to build efficient BCI, what it should be doing is moving, changing, basically, the intrinsic field in the depressed patient in area 25 that Helen has found can treat depression from the pathological state to the desired state in a similar fashion that the DJ was changing the speaker, the music to the speaker, and then the pattern in the grains of sand or the molecules was changing. So the second dimension we're testing now with Helen, and this is what I want to finish the talk with some preliminary results, is that DBS for depression might alter the electric fields and the corresponding state of consciousness. But that's what happens. And actually, they see already that it alters the state of consciousness because they have these behavioral assessments with the Hamilton score, etc., that shows improvement after DBS. So something's changing. There is no, for example, fatigue or slowness in the motor responses. So the data we have come from a recent DBS study, and this is the hypothesis again I put up here, that DBS changes electric fields. So what does it show? I hope you can see, yeah, you can see more the whole slide, yeah. So on the vertical axis here, you have power, and then before and after the DBS. The blue dot is before, the red dot is after. And this second plot, B, has percentage drop in beta before and after, so it has these basically numbers as single points now on the horizontal axis. And then the change in the Hamilton score, what is the improvement in the behavioral assessment before and after is shown on the vertical axis, and there is a correlation. So what does this tell us? It tells us that if we do the following manipulation, we'll see improvement. What is a manipulation? Is putting a deep brain, an electrode in area 25, which is part of the limbic cortex that controls serotonin and is part of emotion regulation. This is the area that Helen had found. You see here, deep in the brain. And we apply a certain protocol that you see on the top right. So 5 minutes we record before stimulation. Then there is 32 minutes of stimulation, another small break of 5 minutes, and then 20 minutes of stimulation. That's what happens in the operation room, right? So there is surgery, there are recordings before the surgery, and then there are recordings with the electrode inside the patient's brain, the depressed patient. And then essentially we measure changes in beta power associated with depression, also in the context of predictive coding with motor commands. And that's interesting, right? Because, for example, in depression and in Parkinson's, we have changes in motor commands, of course, that are among the primary symptoms. And then this drop in beta that we see correlates with improvement in the Hamilton score. So what I want to do now, remember hypothesis 2 that I want to test, is that DPS changes the energy state and the electric field. And of course, I'm omitting some math. I will give you a quick summary of the math we are using, which is basically finding measures of stability and whether the attractor state is the same or different. And mathematically, in the theory of dynamical systems, the way to do that is using, among other tools, something called Lyapunov exponent, and doing a reconstruction of the phase space, basically. So the Lyapunov exponent I try to explain and give a high-level summary on this slide. It characterizes, I hope you can see the red. The red says the rate of separation of trajectories that start or end at equilibria, in other words, states of consciousness. So I have a state of consciousness, and then I want to see whether brain dynamics stay at this state or they are moved. So I'm using this measure, which does exactly that. It takes the trajectory over time, the change in the brain dynamics, and assesses whether it stays at the same equilibrium or diverges. And it does it in a very beautiful and simple way, as all math. I have a PhD in math, by the way, so I like this sort of simplistic or reductionist approaches. So if this Lyapunov exponent is positive, it means that the small differences at the very beginning of the dynamics at initial conditions will grow exponentially over time, and these trajectories, these two different evolution of brain dynamics will diverge. So I will move to a different stable state or a different attractor. But crucially, if it's negative, it means that I stay in the same energy state and I don't move. So if L is negative, the Lyapunov exponent is negative, the system is stable to small perturbations and will return to a steady state or periodic orbit. So if I have brain dynamics before and after, and then as time goes by, as you will see, and it's negative, it means I didn't change. DBS didn't do anything, didn't change my energy state. So this is the preliminary analysis we did, and we found this result that indeed DBS changes the energy state because the Lyapunov exponent that you see here at the bottom switches between blue, which is negative, and yellow, which is positive, different subjects here. So what do we have here? Let me wrap up with this slide. That's my last result. We have the manipulation that you saw earlier, and we take now the patient out of the operating room, and we look at the evolution of brain dynamics for a whole week, and also at behavioral measures. So we have measurements of brain activity without stimulation now. And the question is, why does DBS work? Perhaps it changes the energy state of the patient even when it's off, right? For the whole week after there are some residual effects that lead to treatment. And if that's the case, then the energy state should change, because we said that perhaps depression is an energy state, and going from depression to healthy state would amount to a change in electric fields. And this is what we find here. We find that essentially we live, for example, this is the phase space diagram, where you have an energy state P, let's say the pathological, you want to go to this other energy state Q. So in the phase space, trajectories mean change in dynamics between directions that control the system. It could be synaptic plasticity, it could be white matter changes. You can describe all this with a biophysical model, and that's what we have done. And crucially, there are certain directions that are stable, as you see here, that are yellow, and the arrows point back to the pathological state, P, but then there are these other directions, for example, that arrows are positive, are away from the stable state. They are positive Lyapunov exponents, and they go away and they move to a different state. So if it's positive, like we said, we move to another state, and that's the preliminary evidence we have here. So what you see at the bottom is basically it's basically measurements over 7 days, 2 measurements per day in the morning. So 2, 4, 6, 8 for a week post the DBS treatment. And you see that basically the blue and yellow Lyapunov exponents are intertwined. And there is a correlation between the Lyapunov exponents that assess stability and the beta power that we saw earlier is also correlated with the Hamilton score. So there's a nice link between how good I do behaviorally and whether I stay or leave the energy attractor. So that's my summary. Quickly, we say the suggestion is that consciousness studies are useful for BCI, particularly using EM theory and different configurations of the field correspond to different conscious states. We showed in vivo evidence that conscious states are shaped by electric fields, the influence groups of neurons containing thoughts in a top-down way, and also the cytoskeleton. This we call cytoelectric coupling. We also showed preliminary evidence for in vivo changes in electric field and states of consciousness following DBS. And with this, I'd like to thank Stuart for bringing us all together and collaborators, and you all for your attention. And please get in touch. I would be happy to discuss any of this, even if it sounds a bit too much.
Thank you, Dimitris. That was great. Question? We have time for questions.
Yeah, really good info and encouraging stuff on depression and DBS. And I want to ask you a similar question to what I asked Earl yesterday regarding the comparative richness of causal structure in the spike code versus the field code. We did a rough calculation recently using Ruffini et al.'s velocity calculations and found there's about 100 million times more information content in the effective field effects versus spike code in comparative situations. If that's the case, you know, it's a huge difference of actual impact on consciousness and our function. Does that number sound right to you at all, or does it sound way too much?
I mean, to assess this number, right, one would need to build a model and do, that's what we do, and do a simulation or even fit it the data and fit alternative models. So for example, I didn't show this result here, but to get this directionality, besides Granger causality, we did exactly that. I had this model where there was a Fabry coupling versus a model that there was no, and I compared fitting. So what I'm saying is, for me as a modeler, I can only say what's true, and especially quantitatively, when I have a model of my own. Now I'm trying to think if there is any other intuitive argument. I would say the corresponding intuition would be the level of expertise that perhaps a conductor has versus an individual musician with regard to how an orchestra operates, and I believe there is a big gap. Now, if it's 1 million times or more, or less, I don't know. But the main thing is that if someone needs to know, it should be the field level. That's our argument. If someone has to really have an overview of and a richer information content, this can only happen at the population field level as opposed to individual spiking of a single cell.
Thank you.
Let me make one comment, take the prerogative again. If you're going to interface, it may be easier to go right to the microtubules, which radiate in megahertz, gigahertz, and terahertz. And so you could use ultrasound to access them. And maybe more important, you can read them out with the new device that we've developed that detects megahertz, which is impervious to eye blink, and muscle artifact, which is a big problem for. And there's going to be an exhibit starting, I think, at lunch today, where you can try it on yourself. So it might be something we can talk about.
Sure, sure. My point is, okay, how precise, spatially can this be? I'm sure they have. I'm saying this is a challenge, and I'm sure people have ideas. Yeah, okay. I'll be glad to. Yeah, go.
I think he was there, and then let's go over there.
So,
over there? Okay.
Thanks for a great talk. I particularly enjoyed how you explicitly explained your graphs. So, if you assume that the electric field has something to do with consciousness, a very simple question. If you apply TMS and you find the right position, could you turn off consciousness? I mean, TMS changes the field, right?
And is your presupposition that there is a single...
Yeah, that's the question. Would you suppose that we may find a position on the head? to apply TMS in order to turn off consciousness?
No. And the reason for that is, at least as a person, as a theoretician who talks a lot with experimentalists, is the closest analog I know that we have data is memory networks. And actually, I study memory as a proxy for thoughts and consciousness. I'm not a conscious person. So if you talk to people like Tonegava, for example, at MIT, they have these recordings during memory experiments where they induce, for example, with optogenetics, fear memories or reward memories anyway. And they measure for a whole rat brain, and they find 50 areas where you need to have an orchestra to have a memory. So what I'm saying is consciousness, or at least memory, is the result of many orchestras playing across multiple areas in the brain, 50 areas. So it cannot be that if it's simple memories are like that consciousness is at one location.
Okay. So 50 or more TMS devices, this will work?
If you can find a precise synchronous way of, perhaps you could. Yeah, if you kill the whole brain, yes, I'm sure there will be no consciousness.
Okay, thanks.
Please be brief.
I'm going to,
yeah, I have a question. So the question I have is, have you studied other conditions, things like ADHD and also like psychedelic states on this? And the reason I ask is also a personal question too, where I took MDMA for the first time in a ceremonial setting, and it actually changed my eyesight permanently and activated ADHD more strongly. So I've been very curious about just how to view, why did that happen, and also ways to, hopefully reverse the effects of that. It was like 100 milligrams of it. So yeah, curious your thoughts on that.
I think the top-down effects of electric fields and the control over spiking, I guess, similar to biological phenomena, should happen across many circuits of the brain. Now, if you want to change the energy state in a circuit that relates to a certain disorder, here it was area 25, schizophrenia, it could be frontal areas, ADHD, some other areas, perhaps you can apply the similar kind of idea. The idea is that one way to characterize pathological states and maybe even bringing them back to health states is by changing the aggregate electrical activity. So perhaps, there is a link to that. Now, how it works, it will be many years before we know, I'm sure.
Cool. Yeah, looking forward to more studies on ADHD and also psychedelics. So.
Say it again?
I just said I'm looking forward if you guys end up doing more research on ADHD specifically or psychedelics.
I mean, I personally work on schizophrenia and depression because the grants are also in that field. But I'm happy to talk to people and collaborate in these directions.
Cool, thank you.
Last question.
Hi, thank you for the talk. I was expecting a little more on state dependency problem in neuromodulation, and for example, in this context with the cymatics for the orchestra, there's, as you say, there's all these different songs being played at the same time, different places, and don't you need to be able to get a good baseline, measure the current state of the brain that's non-stationary, and adjust the stimulation to that to have the effects you would want? I think it's the main problem in neuromodulation that no one has a real solution for it.
Yep, absolutely. Yeah, this should be all online, a closed loop. And there are some results. For example, Earl Miller has done some work on that, Alec Weeds also, and others, where basically on the fly, you can control oscillations. So you have this intrinsic oscillation, could be gamma for schizophrenia, for example, or motor impairments, beta, and then you know what the intrinsic resonance, the pathological oscillation is of the circuit, and then you have an external field that is applied and tries to modulate this. And of course, getting rid of delays, right, and doing it on the fly is a big particular challenge, but I think people have already shown results promising this direction.
Okay. Do you think there will be I guess the question is that the crucial piece that will need to be figured out for the success of PCI in the future?
That's definitely very important. What I try to say here and sell to you is that it's sort of the level of description. How do we characterize interactions between our invasive neuromodulation and spiking? That is also important, and we need the right language. And for me, the right language is these two levels that people often forget. They say, okay, spiking, for example, only, but I say it's the interaction between field and spiking.
Okay, thank you.
All right, let's give DIMITRIS PINOTSIS a big round of applause. Thank you very much.
Thank you.
And we'll reconvene at 11:10 for our keynote presentation in the AI world by Susan Schneider. See you then.